Hi everyone, welcome back. In this video, you are going to learn about transport through cell membrane part 2. In this video, we are going to talk about active transport and fascicular transport. Please allow me to remind you on the things that we have discussed in our class. So far, we have learned about passive transport. In passive transport, no energy is required because the substance or molecules moves down the concentration gradient. Very much like the diffusion that happens in the nature without living things. However, active transport requires living things because it needs energy that is expensed by a cell. Why active transport needs energy? Because active transport moves molecule against the concentration gradient. That's why it requires energy. Before we talk about the active transport itself, let's talk about ATP, adenosine triphosphate, which is the batteries of a cell. Now, ATP is a big molecule that consists of three different parts. The first one is adenosine, like the one that you can see here, adenine. We will talk about adenine further when we enter chapter 2, biochemistry. And the second group is ribose. Ribose is a type of sugar that can be found in genetic materials such as RNA. You will learn about it further as well in chapter 2. Now, the last part of the ATP is the 3-phosphate. 1, 2, 3. That's how ATP got its name, triphosphate. Now, as you can see from this picture, from the picture number 1 on the top, you can see that the first phosphate has a lot of energy, while the second phosphate has some energy. And the last phosphate will release a little bit of energy. Now, if one phosphate leaves the ATP, it will become, ATP become ADP. The D stands for diphosphate. So it is called adenosine diphosphate. Now, if some more energy is released, but of course this time the amount of energy less than energy that is released by the first phosphate, the ADP will become AMP. Adenosine, can you guess? Yes, monophosphate, because there is only one phosphate. Now, from AMP to ADP, they can be recharged, all right, by adding additional phosphate group. If AMP is recharged through the cell respiration, it will become adenosine diphosphate. The same thing also happens from ADP to ATP. Through the cell respiration, the last P can be added to ADP and now become ATP, which is a fully recharged cell battery. Okay, now you're ready to learn about active transport. Now, unlike the facilitated diffusion, where there is protein channel, here you will meet with protein pump. Can you see the difference between protein pump and protein channel? Look at the whole diagram from number 1 until number 6. Yes, the difference is in protein channel, the protein remain passive, remain the same. It just facilitates. However, in active transport, you can see in diagram number 3, the protein pump changed its shape. In the first diagram, the protein pump facing inwards, while in diagram number 3, they are facing outwards or exteriors of the cell. All right? Now, let's talk about this process. Let's say this is a green molecule that is found in low concentration. On the outside of cell, the concentration is high. By facilitated diffusion, the molecule will move the other way around, from outside to inside. But here, the cell wants to expel this substance from inside to outside. So here comes the hero, ATP. We have learned in a previous slide that ATP will release one of its phosphate and gives off the energy to the protein pump. And this green molecule will attach to the place where it fits to the shape of the molecule. Because of the existence of energy from phosphate, the protein pump will change shape and then expel the green molecule to the outside. And after that, the other molecule that needs to be actively transported, the orange one, it will attach to the other side that fits to its shape. Now, after that, the phosphate will leave the cell. When the phosphate leaves the cell, the energy is gone, vanished, it has been consumed. As the result, the protein pump will go back to its original shape as in the beginning. Okay, now I will show you a video to help you to have a better understanding of how the active transport pump works. Now you can see here, there is a protein pump, the green one. The green molecule wants to enter, but since there is no ATP, they cannot get in. See, after the ATP attached, the red one, it changes shape. Let's see once again. The green molecule, ATP arrived, yes, and then the ATP shape changed. Now I hope you understand about the active transport. The next is, what if the molecules are so big, thus they cannot pass through the channel or pump? Like in this picture, two people are pushing this big giant elephant to enter this small house. Look at to the door. For sure, this elephant will not fit through the door. So what to do? Yeah, you have to break the wall. We will see the next slide, and it is called vesicular transport. So this is how a big molecule enters a cell. Okay, now, well, it is not literally break the plasma membrane, but at least you can see from this picture that the plasma membrane, part of the plasma membrane, will break apart from the overall plasma membrane. As you can see from here, this is how it works, right? I'll delete it first. Okay, so again, the first step is the plasma membrane will create a curve that is facing inwards, or the cool word for this is called invagination. And then the molecule will enter to this cave, all right? And then the plasma membrane will cover the molecule, and then the two parts, the two ends of this plasma membrane will join. And then at one point, they will separate from the large plasma membrane and create a small bag made of the plasma membrane. This is called vesicle. Now you can see from here, now the big giant molecule is already inside the cell and it is covered by a plasma membrane or vesicle. Now, this is the opposite of endocytosis called exocytosis. In exocytosis, for example, there are huge molecules that the cell wants to get rid of. They want to export it out. So, the first step is the vesicle will move towards the plasma membrane, and then after that, they will become very, very close so that the plasma membrane will fuse. And then, after that, the opening will be created, and then the molecules will be released outside. So basically, exocytosis is the opposite, exactly the opposite of endocytosis. Now, I want to focus on this ATP, even though you cannot see the ATP in the diagram, but it is require ATP because there are some movement here, changes in the plasma membrane. All right. In this video, I want to show you the endocytosis in action in daily life, right? This can be seen under microscope. So in this video, you will see an amoeba that is eating two paramecium, right? Let's see the video. Okay, so this is the amoeba. Now you can see the invagination, the plasma membrane. Yeah, now that's the point where the plasma membrane meet. Now this paramecium is already inside the fascicle. So there are two fascicles here, right? So let me repeat for you. Okay, see? Now this is the place where the plasma membrane meet and enclose the paramecium in the fascicle, right? Transport using fascicle is not only to get rid out of things from the cell or take in big materials from outside of the cell into inside the cell, but fascicular transport also happens within a cell. Now, I hope you still remember in the first subchapter, which is 1.2, if I'm mistaken, that talk about the prokaryote and eukaryote. Now, in eukaryote, there are so many internal membrane system or core organelles. So, for example, can you recognize this? Yeah, this is the RER, reticulum, rough endoplasmic reticulum. So you can see the brittles, the red thing on the surface of this reticulum endoplasmic. So they are the ribosome. Now, if you still remember, the function of ribosome is to produce protein. So in this part, the protein has been created by the ribosome attached on the surface of the RER, rough endoplasmic reticulum. Now, this protein has to be exported into the Golgi body. Which one? Yeah, the green one. Yeah, correct. That's the Golgi body. Now, first, the protein has to be packed inside a fascicle that bud off or leave the reticulum plasmic, and then this will fuse to the membrane of the Golgi body. Right? I'll erase my membrane. Right. So you can see it here. See, the fascicle is created, bud off, and release, and then the fascicle will fuse with the Golgi body membrane. And the function of the Golgi body is to modify the protein so it will become a specific protein with a specific shape to carry out a specific function. Now, after the protein is modified, now it is ready to be exported out, right? So you can see from here. Now, it will also bud off from the Golgi body, and then it will become a fascicle that is ready to do exocytosis. Now, I hope by learning this, you will understand even more the function of protein synthesis that happens in the rough endoplasmic reticulum. As you can see here, that's not for internal usage. The protein will be used for export or outside of the cell. And the last but not the least, now I want to show you exocytosis in your daily life in your body. Now, this is a secretory fascicle that can be found in, for example, salivary gland, your saliva, and pancreas in your digestive system, and your sweat glands. So you can see from here, there are so many small yellow circles, and they are the secretory fascicle. So basically, this is exocytosis. Now, I just want to tell you that now, after you learn many things about cell biology, I hope you now realize how busy your cells are in their daily life. For whom? For you to support your life. So be thankful to them by giving them the best nutrients, good rest for them to revive, and proper exercise to make them healthy. All right? Okay. See you later. Bye. I also would like to thank for these resources that I have used for this video.